Okay, good afternoon, friends. So in the last class, in yesterday's class, we discussed about nitrogen metabolism, wherein we discussed about the importance of nitrogen and the nitrogen cycle as such, wherein we discussed how the atmospheric nitrogen is being trapped by microorganisms and how it is being taken up by the roots of various plants, and from plants how it migrates to animals, and with the death of animals and plants, how the dead organisms get decayed, and ultimately mineralization takes place with the involvement of various soil microbes, and then how the nitrates or the ammonia, the nitrates and ammonia, how they get interchanged. So wherein we discussed four important processes. One is ammonification, nitrification, denitrification, and nitrogen fixation. These four are the important components of nitrogen cycle. So wherein the nitrogen from atmosphere, after passing through various organisms, again ultimately it reaches to atmosphere from where it has come in. So this cyclic movement of nitrogen we name as nitrogen cycle. And of course, apart from this, we also discussed about the uh, nitrogen fixation mechanisms. So nitrogen fixation, only microorganisms are capable of converting this, and this conversion of an elemental nitrogen into nitrate form or ammonical form, or more specifically ammonia form, we call nitrogen fixation. So this nitrogen fixation in nature occurs by three means. One is, physical nitrogen fixation, chemical nitrogen fixation, and the third one is biological nitrogen fixation. So physical means which occurs naturally. So in nature, when the weather becomes very rough during lightning and thunder showers, and during that time, under high pressures, under high electric discharge, the nitrogen atom, a stable nitrogen molecule, splits into nitrogen atoms, and these nitrogen atoms may fuse with uh, oxygen and give rise to various oxides, nitric oxide, nitrogen dioxide, and which may combine with the water vapor, and of course HNO3 or nitric acid formation takes place, and this reaches soil, and where it fuses with the various metals and organic metals, and ultimately ultimately give rise to what we call their salts. Then this is naturally occurring. But of course, the fixation that occurs in nature is, when compared to other processes, is a bit less. And the second is, of course, a chemical process wherein uh, this nitrogen fixation mechanism is being done in fertilizer factories, wherein under high temperature, as high as 450 and 470 degrees centigrade, and a high pressure, maybe around 1000 bars of pressure. Under such extreme conditions, the nitrogen molecule breaks into atoms, and these atoms of nitrogen fuse with hydrogen, and of course we get ammonia. This is what the principle that is behind industrial nitrogen, or our fertilizers. The third is biological nitrogen fixation. Of course, this biological nitrogen fixation, as already said to you, this is done by microorganisms, and these microorganisms are primarily of two types. Of course, some are free living, others live in a symbiotic relationship. Free living mostly are, cyanobacteria or blue-green algae. A good number of cyanobacteria, blue-green algae are known to fix atmospheric nitrogen, thereby enrich these soils, especially the rice growing areas where there is a water, stagnant water will be there. On that you will see these blue-green algae growing on the surface of the water in a floating manner, and they fix atmospheric nitrogen. And of course, the other group is the bacteria, and these bacteria are some are free living, and other are symbiotic. Free living, they are again anaerobic, aerobic, etc. Of course, the aerobic is Azotobacter an example, and Clostridium is an example for anaerobic bacterium, and photosynthetic or Azospirillum, etc. And the most important ones are symbiotic nitrogen fixers. The symbiotic nitrogen fixers are mostly Rhizobium species. Rhizobium brandi, Rhizobia species, they are capable of fixing this atmospheric nitrogen when they are associated with the roots of various legumes. And they form what we call root nodules. And of course, I told you, and the important enzyme that is involved in all biological nitrogen fixations is nitrogenase. Nitrogenase is an enzyme that is involved, and this nitrogenase enzyme, is very sensitive to oxygen. If it is in a oxidizable condition where oxygen concentration is more, there as it is very sensitive, it breaks, it dissociates, it denaturates, thereby it loses its ability to fix nitrogen. So in nature, or in the root nodules, or in the heterosis, there is what we call compartmentalization, and there is what we call in root nodules, you have a novel pigment by called leg hemoglobin, that regulates the concentration of oxygen in that vicinity and thereby facilitates. So this is, of course, about nitrogen cycle. Of course, apart from this, we also learned about what we call the amino acid synthesis. So ammonia, after fixing, it has to enter. It will be taken up by acids, carboxylic acids, and these carboxylic acid, the entry, they take up this ammonium and or ammonia and convert themselves into respective amino acids. The formation of amino acids is by two means, what we call transamination and reductive amination. Of course, we read about this. Transaminases, generally they transfer amino group 1 from one amino acid to the other, or from one keto acid to the other. So thereby there is a transfer. As it helps in transfer of amino group, they are called amino transferases. Of course, the reductive amination, where a new synthesis of amino acid takes place. This by two means, what we call, there are two enzymes involved in this, what we call reductive, wherein reductive amination, and other is a GOGAT. These are the two enzymes that are involved in the conversion of ammonia, or ammonia gets labeled or taken in, and ultimately the acid gets converted into an amino acid. Of course, this we have learned in our yesterday's class. So now today, the other important topic of today is plant growth. The meaning of growth and development. Of course, the chemicals are the factors or the chemicals that are involved in regulating growth in plants. That is, we learn about the meaning of growth and growth regulators, and also we learn about physiology of flowering. So this we will cover in the next 50 minutes or so. So now, to begin with, what do you mean by growth? Say, growth is a fundamental phenomenon, and which is experienced by all living organisms. All living organisms, starting from your bacteria to the human being, every organism in its lifetime, uh, gets involved in growth. Okay, so growth, a definition of growth, very difficult. Still, growth is a permanent and irreversible change involving in a change dealing with or affecting uh, uh, in length, width, or volume, or number, etc. So what you have to remember here, it is an irreversible and a permanent change. It is an irreversible and permanent change. These two are very, very important. So it is an irreversible and permanent change. So a growth has occurred means, so it cannot come back. It is a permanent change, irreversible change. And the change involve various, various parameters. So the important parameters are, for example, it may enhance the length. It may enhance in width. It may result in enhancing the volume. It may enhance its enhancement in cell number, cell size, etc. So these are all permanent changes. So this is what we mean by growth. So growth, growth, when starting from a single organism, say a single cell, for example, zygote is the single cell from which it is a single cell organism, of course, it may undergo, in a diploid cell, of course, it expands and becomes an adult. If it is an haploid, so also. But if it is a haploid and diploid, of course, therein you will see what we call reduction division takes place. So whatever the case may be, so in case of unicellular organism, the growth here means, of course, because it is a unicellular organism, it is a bit small. When it reaches its maturity, it may little expand. But here the growth means increase in number. Increase in number. See, for example, in all cases, for example, in multicellular organisms, including human beings, there is both an increase in number, an increase in size, increase in length, increase in volume. So all these, put together lead to what we call growth and development. So now the growth, if you observe any organism for that matter, even including our own human beings, see, we start when we are in mother's womb, our life starts as a single cell, that is zygote. This zygote undergoes repeated mitotic divisions. As a result of that, the multicellular, tissue systems, organs, and ultimately the individual. So all organisms start their life as a single cell, and it is because of cell division and cell expansion and cell maturation, ultimately morphogenesis, leads to a developed individual. Okay, so now growth will not take place at the same pace, at the same rate. The rate of growth may be starting from a cell or a tissue or an organ, a tissue, whatever the case may be. So the rate of growth is not always at the same pace. So if you plant, if you plant, the time and increase in either length or volume, etc., on X and Y axis, you will see that you will be amazed to see that the rate of growth is not same. So initially, for example, take the example of human beings. See, as a child, of course, our growth is there. But when you reach, teenage, there is an exponential growth. Suddenly, you will see an enhanced growth. But of course, after teenage, after 25, 30, of course, then it becomes slow. And of course, the whatever the height we have reached, but even till death, that cannot be there. Of course, you may bend, you may look a little bit short, but actually your height, it remains same. So, what I mean to say is the rate of growth is not always progresses in the same speed. So, uh, say for example, initially the growth rate is a bit slow. For example, if it is an organism or if it is an individual, say for example, if we are there, if you go to a new place, generally we take some time to adjust. After adjustments, of course, you start working. So also cells, if they are kept in, for example, maybe in a controlled condition, or for example, a seed, a seedling, etc. Initially, the growth is slow. This phase we call lag phase, lag lag. Later on, after adjustment, it progresses at a faster rate, so that this enhanced we call log phase, lag phase, log phase, and after some time, then it becomes a plateau, a stationary phase. So lag phase, log phase, and stationary phase. So lag phase, and exponential growth, and then senescence or stationary. So you get what we call, if you plot this time and rate, you will see S-shaped curve. This S-shaped curve is what we call. So this, as far as exam is concerned, this is important. Sometimes they may ask you to write briefly about S curve or growth curve. The otherwise called growth curve. So this, what I mean to say is, initially it is slow, later on it picks up, later on again it remains same, or at times it falls. Say for example, we are there. So if our growth is influenced by many factors, so of course, if food is available, grow. But after accelerated growth, then food becomes a limiting factor, availability. So then of course, your cell division and cell expansion falls. Hence, a plateau. So this is what we call S-shaped curve. So now, then we move to what we call growth regulators. See, of course, we have growth regulators. Even human beings also, we have a number of hormones in us. Pituitary hormone. Pituitary is adrenal gland. Your sex glands. These are all glands that regulate various biological systems in our body. So they are called, what we call, these hormones are endocrine glands, are ductless. They don't have duct. That means they are produced at some place and they get translocated to other place where they show their response. So generally hormones, the place of their origin and the place of their action are separated. This is a hormone. And to differentiate the animal hormones with the plant hormones, hence we use plant hormones, otherwise called phytohormones. And these phytohormones, these hormones are very, they have certain characteristics, and generally, they are organic in nature, and generally they occur in nature, and they show mobility within the plant. So generally, they are produced at the stem tip or root tip, or on the young leaf. These are the areas which where the hormones are produced or manufactured. From there, they get translocated to the site of their action. So they are organic in nature, natural occurrence, mobility within the plant, and they are very active in very low concentrations. Even at minute concentrations, they exhibit their response. And of course, they influence your growth and development. Now, these hormones, for our convenience, these phytohormones uh, are otherwise called, plant growth regulators. Plant growth regulators, otherwise called. And these growth regulators, for our convenience, we divide them into two categories. One is growth promoters and the other is growth retarders. Growth promoters and growth retarders are growth inhibitors. Retarders or inhibitors are one and the same. So growth promoters, that means they accelerate growth, they promote growth. Hence they are called growth promoters. And these growth promoters, mainly the auxins, gibberellins, and cytokinins. These are they fall under the growth promoter category. And of course, the other is growth inhibitors or growth retarders. They are abscisic acid and ethylene. So you should remember them. So this is the brief, meaning of growth regulators. Now we will study their occurrence, their physiological effects, and how they are useful in our day-to-day life, especially in horticulture and agriculture. So our aim is to how best we can make use of these phytohormones in order to enhance agricultural yields. So to begin with auxin. So auxins, auxin was the first, phytohormone that was first discovered, and its physiological effects were first studied. And of course, in Greek, this auxin means the material that increase. Auxin means increase. So that means it increases the activity in a cell, in a tissue, in an organ, or in a plant body as such. So they are increasers. So they increase the length of the plant body. They accelerate the growth of the either a seedling or a branch or a stem as such. So now the first it was Julius Van Sachs who first noticed this. Of course, before that, even the credit for first observing the effect of the auxin goes to Darwin and his son on Canara grass. So they said that when the Canara grass was exposed, when the coleoptile or what we call stem tip was exposed to a unilateral monochromatic light, generally the tip or the stem tip showed curvature towards sunlight. This is what we call phototropic. So it tried to grow towards light. So based on that, he said that they concluded that there is some substance that is produced in the stem tip that is under its influence only the plant is able to grow towards light. And of course, later on, so many people worked on this. It was, Boyson and Jensen. It was Wendt and Boyson and Jensen. They finally, extracted that, organic compound from the stem tip or from the coleoptile of Avena sativa. And after doing certain experiments, they concluded that there is some, chemical substance that is produced at the stem tip, and to stop the growth, when they cut the top coleoptile region or the young stem tip, and then when they observed, the growth got arrested. And when they placed again the cut portion on the stem tip, again it started growing. This shows that there is something is there at the stem tip that is responsible for growth. And Wendt and others, they put this cut portion on a agar block, and when the agar block was placed on the stump, that is cut portion, the stem showed growth. It shows that there is some material that is secreted in the stem tip. When it was cut and placed on an agar block, that percolated, that leaked into the agar block. And when the agar block was placed, again, it started growing. And through these experiments, they proved that there is some material And this material, of course, later on, so many people worked on this. And finally, they said that they named that substance that is responsible for increase in growth. So that is what we call indole acetic acid. That is what is called indole acetic acid, a naturally occurring phytohormone or auxin. And later on, indole butyric acid was isolated. And later on, because of the uh, initiative of various biochemists, plant biochemists, agricultural scientists, they have produced materials that are similar to auxins. They have same effects. They have same effect on plant growth, and they are called synthetic auxins. And synthetic auxins are, for example, naphthalene acetic acid, 2,4-D, 2,4-dichlorophenoxyacetic acid, 2,4-5-T, 2,4-trichlorophenoxyacetic acid. These are the synthetic auxins. So both natural auxins and synthetic auxins are known to enhance the length of the stem. But of course, not only length, they are known to, increase influence even the root. But scientists, after careful observation, found that the concentration of the auxin that is required for the stem growth, if the same concentration is applied to the root, instead of promoting, it arrested. So it shows that each and every part has its limited, influence on that particular plant. Generally, stem needs more concentration of auxin, whereas root needs little concentration of auxin. Through all these experiments, they found that auxin is very, very essential for the growth of both root and stem. Okay, so now this auxins, auxins show what we call when the agar block was placed, instead of placing on the stump, when it was placed on one of the edges. Generally, when it was placed on one of the edges, the other side, the opposite side, if you put like this, generally opposite side showed curvature. That means the whatever the auxin that percolates, that gets translocated, there it enhances cell division and cell expansion. Thereby, the stem shows growth in an opposite direction in the form of a curvature. So based on the amount of curvature, we make what we call bioassay. Bioassay is nothing but it is a kind of observing determining the efficacy or the strength of the substance. It is a phenomenon of what we call the testing the efficacy and strength of a substance when it is tested with the help of a living material. We call this as what we call bioassay. So by doing various experimentation, the bioassay of auxins were done, and they showed, when it was exposed to sunlight, generally, because it is very, very sensitive to sun, so it moved to shade part. So with the increase in cell, division and elongation in the dark phase, in the darker part, it showed towards light, phototropic movement. So also when it was placed in a eccentrical way, and of course it showed the opposite side growth and making a curvature. So based on all these things, they concluded that auxins show phototropic movements and also geotropic movements, and they also show what we call thigmotropism. Phototropism towards light, and movement towards geotropism. Root show geotropism. Generally, stem shows phototropic movements. So phototropic and geotropic, of course, in case of cucurbitus, etc., they whenever anything that support comes, it touches. This is what called thigmotouch, thigmotropism. So these are the tropical movements. Now coming to the physiological effects or applications of auxins. Generally, the apical dominance is one. Apical dominance means generally, the suppression of the apical bird, suppression of apical bird, the lateral buds. So the apical bud suppresses the growth of lateral buds is called apical dominance. So generally, what I mean to say here, see, for example, Eucas polyalthia longifolia, what we call ashoka tree, or eucalyptus, or pines, etc. They show lengthy movement, growth, and generally the branches are very limited. So they suppress. The apical bud suppresses the lateral bud's growth. So this results in a conical elongated growth, long conical elongated growth. This is under the influence of what we call the auxins. When you detop, when you cut the apical bud, now you will see within no time the bushy growth. That is, lateral buds become active and plant becomes a bushy growth. So this phenomenon has been used in many plants. For example, in case of tea plant, if it is bushy, you can see many branches and you will see many tender leaves, which give you quality tea. Of course, if it is a hedge plant, uh, as a border plant, if a plant grows tall, it is not useful. If it spreads laterally, that is good. So in case of hedge crops also, the top is removed so that the lateral branches develop. This is one phenomenon of apical dominance under the control of the auxins. And the second one is rootings. Those stem cuttings nowadays, for example, in case of rose, etc., you cut the stems, and if you dip them in auxin and if you plant them, you will see the root initiations fast. And the third one is parthenocarpy. See, parthenocarpy, as you know, it is development of the ovary into a fruit without fertilization. This is called parthenocarpy. So in some cases, the fertilization does not take place till still the ovary develops into a fruit. And because no fertilization has taken place, no seeds are there. So even if seeds are there, generally they don't have any embryo. So they are called seedless fruits. For example, your grapes or oranges, even certain melons, if they are seedless, we prefer them. So in getting, seedless fruits, if you spray auxins, you get parthenocarpic fruits. Then, of course, the other important physiological effect is in case of, they also show what we call, uh, they are used to get rid of what we call, weeds. They are used as weedicides. The auxins are used as weedicides. Say, for example, weeds, nowadays the labor costs have gone up. So the 2,4-D and 2,4,5-T, they are used as weedicides, especially in rice crop. Rice is a monocotyledonous crop, and in that, any dicotyledonous crop, any dicotyledonous plant developing in a rice crop becomes a weed. See, so by spraying these weedicides, especially the auxins, you can get rid of these dicotyledonous plants. So also in case of pastures or in case of our grounds, the ground maintenance, the green grass maintenance in case of gardens is done by spraying these. They check, they eliminate these unwanted dicotyledonous plants. So these are, some of the important, physiological functions of auxins. So then of course, in cucurbits, the other important effect has been found to be the cucurbits, generally in case of, bottle gourd or ridge gourd, wherein you will see the flowers. There are two types of flowers, male flowers and female flowers. So if you spray these auxins, they promote the female flowers. So thereby, if female flowers grow, you get fruits. So thereby, but this application is of economically very, very important. This is about the physiological and effects of what we call auxins. Then we move to gibberellins. Gibberellins, another group of phytohormones. And there is a story, you might have heard, uh, gibberellins, foolish seedling growth. Foolish seedling growth, wherein Gibberella fusicori. It was first observed in a rice field by a Japanese scientist, and it was Shumakura who first noticed or made a thorough investigation and found that the foolish seedling or extra length of the seedling of rice was because of the infestation of the roots of that by Gibberella fusicari. And this Gibberella fusicari, a fungus that produces gibberellin. And under its influence, the plant grew very fast. So it enhances growth. It is also another important, growth promoter, gibberellic acid. And there are, of course, there are many gibberellic acids are present. They are named, GA1, GA2, GA3, like this. There are 30 to 40 gibberellic acids are there. All gibberellic acids have their own, effect on growth in plants. Of all, gibberellic GA3, gibberellic acid 3 has been very, very popular. Now, the importance, the importance of this has been, they are known to increase in length, especially the what we call sudden increase in internodal length. Sudden increase in internodal length, what we call bolting. So in many crucifers, like your cabbage, cauliflower, etc., sudden increase in the internodal before flowering is called bolting. And gibberellins are known to enhance bolting processes. And of course, we have in many crops, there is what we call genetic dwarfism. So if a gene gets modified, and the gene that is responsible for length, if it gets modified, the plant becomes dwarf, short. And this genetical dwarfism can be overcome by spraying these gibberellins. So removal of genetic dwarfism can be done by spraying these gibberellins. And of course, bolting and flowering, I told you. And seed growth, if seeds and seedlings, if you spray this very low concentration of gibberellins, they are known to enhance seedling formation or seed germination. Seed germination gets increased. So the gibberellic acid is known to interfere with hydrolysis of reserve food material. So when food material gets oxidized at a faster rate, of course, energy availability gets increased, and under its influence, the cell division, cell expansion increases, and thereby, seed germination also gets hastened. Now, the parthenocarpy, like auxins, even gibberellins are known to help in parthenocarpic fruit formation. And of course, unlike auxins, these gibberellins are known to favor male flowers in cucurbits. Of course, this is not an advantaged, not an advantage factor for the, especially for the farmers. So here, especially the plants growth can be enhanced, especially in soybean and others. And of course, dwarf plants can be, made to grow to a normal size, bolting. And of course, de novo synthesis of amylase during germination are known to, and they have a role, they are known to check to certain extent the apical dominance also. Then we move to cytokinins, another third category of phytohormones. And it was Haberland. It was Haberland, and later on it was Skoog and Miller. They isolated this cell division promoting hormone known as kinetin. And this kinetin was, of course, later on, it was initially isolated from herring sperm, and later on it was isolated from Gia maize and also from coconut milk. And from that, whatever the cytokinin that was extracted was known as kinetin. And this, as the name indicates, cytokinin that helps in cell division. So especially if you are doing what we call tissue culture, therein if you add required concentration of cytokinins to the callus, it helps, it enhances cell division, and also it is known to enhance morphogenetic process also. So this hormone cytokinin is primarily, it regulates cell division, and of course it is an adenine compound. It is an adenine, a purine a derivative it is. Now cytokinins are synthesized mostly in root system. Cytokinins, unlike auxins, which are synthesized in roots and shoots, but cytokinin formation mostly is restricted to root system only. So now physiological functions, cytokinins, especially as the name itself tells you, it helps in cell division, cell elongation, and also morphogenesis and organ formation. I told you, so this can be profitably used in tissue culture experiments. So especially the cytokine and auxin ratio, if it is 3:1, generally it promotes shoot differentiation. And if its concentration is, if auxin concentration is more, and cytokinin, the cytokinin concentration is less and oxygen concentration is more, it is known to increase rhizogenesis. So the what we call collagenesis and rhizogenesis, for them the concentration of cytokinin and the oxygen ratio varies. So if cytokinin ratio is more, it promotes collagenesis. If oxygen concentration is more, it promotes rhizogenesis. Of course, it is known to what we call delay senescence. So senescence is yellowing of leaves. As the plant grows old, generally you will see seasons also, there is shedding of leaves. So this yellowing of leaves is called senescence. So generally, if you spray the cytokinins, they are known to delay senescence. So this process, this has been, so the ability of the cytokinins to delay the process of senescence is called Richmond-Long effect. Richmond-Long effect. Suppose, so this can be used, for example, in case of leafy vegetables, your shelf life of the leafy vegetables and also shelf life of what we call fruits can be enhanced by using these phytohormones. Of course, stomatal opening, cytokinins are known to promote the stomatal opening. Then, so these are the three: oxygen, gibberellins, and cytokinin. They are promoters. And then we move to what we call growth inhibitors. Under this category, we have two, of course, abscisic acid, and the other is ethylene. So abscisic acid is its effect was studied by three people separately, and of course, three people named them differently. One as abscissin 1, the other abscissin 2, the other man named it as dormin. So like this, they were variously named, like abscisse, abscissa 1, abscissa 2, and dormin. Later on, when all their research papers were studied together, it proved that whatever the name they have given for the growth retarder was one and the same, and it was renamed as abscisic acid. And this abscisic acid is a terpenoid, a sequiterpene of 15 carbon length. Sequicorpene is our abscisic acid. So this abscisic acid, it helps in what we call abscission. Abscission is nothing but separation, breakdown. So your fruits or leaves, when they mature, they fall. The separation of the fruit or the leaf from the parent part is because of the development of special tissue called the abscissin. The abscissin separates. So thereby, leaf or the fruit falls from the parent plant. So this is what we call abscissin. So abscissin along with the ethylene are known to enhance the process of this senescence and abscission. Then dormancy. Dormancy is temporary sleeping. We have so many plants, so many seeds, generally after their formation, some seeds are able to germinate immediately. But some, because of the immature embryo, they need some time. So soon after their formation, even if a plant also, they don't germinate. Why? Because the embryo is yet to retain, yet to mature. So that is how. And in some cases, for example, in case of legumes, you have hard seed coat. The hard seed coat does not, permit the entry of oxygen or entry of what we call water. Thereby, it checks imbibition, and thereby it checks seed germination. So if you put this abscisic acid, you can enhance dormancy. See, for example, in some cases, we have to break the dormancy. For example, if you breaking of dormancy can be done by exposing to gibberellic acid. But exposure to abscisic acid, it enhances, the dormancy. So abscissin enhances and dormancy. For example, in certain cases, we want to, enhance dormancy. For example, you buy potatoes. If you keep in our house, potatoes, they sprout. When sprouting occurs, they are spoiling. If you check this buds, by enhancing dormancy, that is an advantage. So this dormancy, enhancing dormancy in certain cases is a profitable thing. So generally, abscisic acid is known to enhance this dormancy. And even the stomatal movement, you know, the cytokinins are known to enhance stomatal opening, whereas the abscisic acid is known to close stomata. So thereby, if there are stress conditions, if there are drought conditions where there is a scarcity of water, so plants, they themselves, as a protection mechanism, they increase the production of abscisic acid, thereby they close. So even farmers can spray. If there are drought conditions, farmers, if they spray this abscisic acid on their farmland, they make, they conserve whatever the little moisture that is there in the soil. So this can be used as as an artificial anti-transparent, as an anti. So this is, and even in some cases, perinating buds. So buds can be perinated by spraying this abscisic acid. Then finally, ethylene. So unlike all the four, the ethylene is a gaseous form of hormone. The ethylene is a gaseous form of hormone, and it was first, the credit for identifying this ethylene goes to H.S. Cousins in 1910, and he said that he he was the first man to say that don't transport oranges with bananas. He observed that oranges are known to enhance the ripening in bananas. Why? Because the ripened oranges are known to release this gaseous form of ethylene, and that enhances what we call ripening of bananas. So the ripening hormone, what we call, is ethylene. And nowadays, a lot of, if you read paper, this is the season, wherein you get mangoes. And nowadays, people are using calcium carbide or ethafone, etc., as artificial ripeners. So ethafone is a chemical that releases ethylene, thereby it hastens the ripening. So this is a ripening hormone is ethylene. So now, in addition to this, this ethylene is known to help wound healing. In case of plants, if there is a wound, it is known to help in uh, healing of the wounds. The other important functions of ethylene are it inducts root hair formation, and of course it increases uh, like our auxins. They are known to increase female flowers, pistillate flowers, and they are known to accelerate abscission in leaves and flowers, and they reduce apical dominance. Generally the apical dominance, this can be to certain extent checked both by cytokinins and also ethylene. But of course, in aquatic plants, they are known to promote aerenchyma so that they float. Now, physiological functions, of course, fruit ripening hormone, and there is what we call triple response growth. This triple response growth is what we call, it inhibits elongation, it stimulates lateral growth. Lateral growth, that means it checks what we call apical dominance. So it inhibits elongation, it checks lateral growth, and induction of transverse geotropism. So ethylene is known to affect these three phenomena. Hence, it is called a triple response growth. And generally, in several uh, plants, it delays, it suppresses, it delays flowering. So in many cases, it delays flowering. So these are some of the responses, and of course, we will study in all together. So how these growth regulators can be used in agriculture and horticulture. See, for example, first, uh, auxins, etc., can be used in stem cuttings for fostering of their fixation and faster formation of root hairs and lateral roots. And weedicides, I already told you, 2,4-D auxins, 2,4,5-T are known to use as weedicides. And apical dominance, of course, this can be removed by detapping and also by spraying the other whereby lateral buds growth can be rapidly enhanced. So especially this apical dominance, of course, used in what we call pruning gardens, and also in tea gardens, and also in hedge plants. Then breaking dormancy, especially gibberellins are known to break dormancy. And of course, the genetically dwarfs can be promoted to reach a normal height by spraying gibberellins, of course. And this abscission of flowers and fruits, is controlled by auxins. But of course, the abscisic acid abscissin enhances, and this can be checked by spraying the untimely drop of uh, fruits, unripened fruits and buds can be checked by spraying auxins. And of course, parthenocarpy, both auxins and gibberellins are known to promote parthenocarpy. And seed germination, of course, the uh, gibberellins and also cytokinins are known to help the seed germination. And shelf life, this is very, very important. You have to make it a point. Generally, if you buy, leafy vegetables or fruits and vegetables, etc., if they remain fresh for longer duration, it's an advantage. So by spraying, cytokinins, especially benzyl adenopurine, and you can enhance the spinach, lettuce, asparagus, etc., for longer duration, and also the enhancing vase life of flowers. The flowers can be kept afresh for longer duration. Example, the poinsettias, dianthias, etc., can be kept afresh by spraying, the benzyl adenopurine. Benzyl adenopurine is cytokinin. Cytokinin generally they enhance the shelf life of flowers and vegetables. So now then the what we call so in another 5 minutes I'll complete. So photoperiodism. This is another important thing. So this photoperiodism is nothing but the relative influence of day length or day and dark period length, day and night periods effect on flowering is called photoperiodism. And this photoperiodism, based on the photoperiodism, plants are classified as short day plants, long day plants, and day neutral plants. For example, short day plants. Short day plants means they require shorter day. That is, the night period should be more and day length should be less. So less than a critical period. So such plants which prefer short days and long nights are called short day plants. And examples for them is, for example, our chrysanthemum, xanthemum, and tobacco are some of the plants that grow well in short day conditions. They flower in short day conditions. And of course, long day plants are the plants that require longer days, maybe more than 8 to 9 hours of day length is needed for them. And shorter nights and longer days are very much preferred by long day plants. Examples for them are, for example, radish, spinach, beetroot are some of the examples that fall under what we call long day plants. And there are certain plants, for them, the flowering is immaterial. Whether you plant them in short day conditions or long day conditions, they are called day neutral. Example for that are your cucumber and maize. So hence, the cucumbers and maize, they are available throughout the year, throughout the year. See, for mangoes, there are certain fruits, you see them, of course, because of enhanced shelf life, they are available nowadays. But mangoes you get only in summer. Sitafal you get only in winter. So at a given season, certain seasonal fruits are available because of this day length and um, day length and day night length and day length. So now uh, last one is what we call the photoperiodism. The flowering is influenced by day length. I told you day length or day or night length, for example. So it was people Borthwick and Hendrik working on thoroughly on the phenomenon of photoperiodism. Photo means light. So the period of light, how it influences flowering in plants is photoperiodism. So it is the length of the day and is the length of the night that determines flowering, whether a plant to flower or not. If long day plants are exposed to short day condition, they fail to flower. If short day plants are exposed to longer days, they fail to flower. But day neutrals, they immaterial for them. They flower whether exposed to long days or short days. So now the reason for this, the flowering was studied in detail by Borthwick and Hendrik. They finally concluded that there is a pigment. The pigment that is responsible for flowering is called phytochrome. Phytochrome is the pigment that is involved in the flower initiation. It was in florigen. Some people call it as florigen. It is phytochrome. So the phytochrome has two parts, a protein part and a non-protein part. Non-protein part is called chromatophore. And this phytochrome exists in two forms, PR form and PFR form. So PR form is active in 660 nanometers light, and PFR form is active 735 nanometer light. So PFR and PR are interconvertible. If plants are exposed to bright sunlight of 660, then PR gets converted into PFR form that promotes long day plants. And the short day plants, this PFR form, passively during nights gets reversed to PR form that promotes flowering in short day plants. So this a phytochrome that is actually responsible for flower. And the last part is vernalization, wherein not only the light, but the scientists concluded it was Lysenko. It was Lysenko, a Japanese scientist, who found that not only light, but even the temperature also regulates crop growth and yield. So, we have so many varieties. So, especially in cereals, especially in wheat, rye, and barley, etc., we have varieties. Some varieties are winter varieties, the other are spring varieties or summer varieties. So, see, for example, summer varieties are grown. If you sow them in summer, of course, they flower. If you grow winter varieties in summer, they fail to flower. So the reason for that was it was Lysenko who said that even the winter varieties can be used as spring varieties provided you expose them to pre-chilling treatment. So this pre-chilling treatment of seed or the varieties or the seedlings is called vernalization. So this vernalization is known to influence the flowering and yielding. So the varieties, winter varieties, can be profitably grown in summer, provided you expose them to pre-chilling. So the pre-chilling conditions vary from plant to plant. Of course, some may need what we call 1 to 2 degrees centigrade, and some others are to be exposed to 1 to 10 degree centigrade. So this is the pre-chilling treatment, otherwise called vernalization, also regulates the plant growth and also flowering. This is what we call vernalization. So this is about your what we call growth, physiology of the hormones and their effects on how the various growth promoting hormones can be profitably used in agriculture and horticulture, and the phenomenon of photoperiodism and vernalization. So now, 2 3 minutes for you to raise your doubts, if any. Okay, if you don't have any doubt, we will call it a day. And of course, we will meet, on second, as far as the timetable that has been circulated so far. Your last class is on the Friday, same time, that is 3 to 4 p.m. And of course, we lost 2 classes. We are yet to get whether they will be compensated or not. If not, your last class will be on the next Friday, wherein I'll briefly explain. If it is a single class, I have to rush through, and I'll explain the meaning of ecology and the ecosystem, ecological succession, and also environmental pollution. Of course, this pollution, of course, I need not tell. This is a well-known concept nowadays. Everybody knows that pollution, water pollution, air pollution, land pollution, etc. You know how to check that. And of course, ecosystem concept also is also known to you, wherein the organisms, the relation between organism and the environment, of course, and ecological succession also. We'll read the succession beginning in water called hydrox succession, and the succession begins on a land, what we call on a rock, we call it as a Xerox succession. Okay, if you don't have any doubt to raise, now I'll close my lecture. Thank you for your patient hearing. Thank you.